In this video, we will discuss about N-Glycan processing, which is a post-translation modification. Though we have covered the N-Glycan processing in one of our previous videos, however, we have not covered uh, in that much detail that we will cover in this video. In this video, we will not only cover which part of glycosylation and glycosylation occurs in which region of the cells. We will also cover what are the enzymes that are involved in forming which kind of bond, and the enzymes are responsible for attaching which kind of monosaccharides. So we will start with this dolicol phosphate, and this is the precursor, glycan precursor, that is formed within the endoplasmic reticulum, that we have already known, and I have covered this thing many times in our previous videos. And after that, with the help of this oligosaccharyl transferase, it is then attached, the oligol phosphate and attached to the asparagine amino acid of the translating protein within the endoplasmic reticulum. Thereafter, with the help of this GlycNAc and GlyC2 within the endoplasmic reticulum, this glucose monosaccharide will be trimmed off one by one till that if the protein acquires their native structure, it will have this kind of monosaccharides, and it will then move into the cis part of the Golgi, that is shown here. And in the cis part of the Golgi, this enzyme will act on this carbohydrate, decorated carbohydrates on the proteins, that is alpha mannose 1, and it will trim off further mannose. Right now, when it enters the Golgi, it will have 8 mannose, and it is 1 2 3 4 5 6 7 8 mannose, and the alpha mannose will trim off some of the mannosidos and leave finally 1 2 3 4 5 mannose. And thereafter, this GNT1 enzyme will act, and it will leads to the formation of this beta 1 2 GlycNAc at this alpha 1 3 bond. And thereafter, this FUT8 enzymes will take place, and it will leads to the formation of this asparagine at alpha 1 6, this not asparagine, this fucose bond formation at this first GlycNAc from the asparagine, and forming a bond of alpha 1 6. And after that, this alpha mannose 2 will trim off further to mannose monosaccharides and leaving just 3 mannose and 3 GlycNAc along with the one fucose structure. And thereafter, this GNT2 enzymes will lead to the formation of this beta 1 2 GlycNAc at the alpha 1 6 bond linkage of the mannose structures. Thereafter, it can go into the different kind of structures based on the enzymes it comes to interact with. For example, and this, if the proteins interacts with GNT3 enzymes, and these things depends on the many factors. Some of the factors include like what are the final destination of the protein based on the signaling sequence, or what are the kind of carbohydrate structures that it has acquired. So these are just some of the factors. There are other several factors also that determines which the carbohydrate structures interacts with which kind of enzymes. The focus of this video is just to know whenever it interacts with a certain kind of enzymes, that will leads to which kind of bond formation. So if it interacts with this GNT3, it will leads to the beta 1 4 bond GlycNAc formation as this middle mannose structures. And if it interacts with GNT4AB, it will lead to the formation of beta 1 4 GlycNAc structures at the side mannose structures, that is mannose that forms alpha 1 3 linkage with the other mannose. And if it interacts with GNT4C, it will lead to the beta 1 4 GlycNAc structures formation at the alpha 1 6 mannose at the alpha 1 6 mannose structures. And if it interacts with this GNT5, it will leads to this beta 1 6 linkage formation at the alpha 1 6 mannose structures. So this is how the different kind of structures forms based on the interaction with the different kind of enzymes. And after that, if it interacts with this beta 1 4 galacty, it will leads to the galactose structures formations, or the glycan sites. That is beta. It will leads to the beta 1,4 beta 1,4 addition, beta 1,4 galactose addition, and both the glycan sites. And thereafter, the silyl transferase enzyme will act. That will lead to this alpha 2,3 sialic acid spawn formations at the galactose structures that is formed in the previous step. So I hope you got an idea how the basic idea how the N-Glycan processing occurs within the mammalian cells. And if you don't understand in detail about the N-Glycan processing, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.